start the session with my presentation on hybrid methods. Mainly, I'll be talking about protein crystallography and Cryo EM, and also I'll be touching upon the allied subjects as well, computational methods, NMR, and quickly on SAXS maybe at the end. So, first of all, before I start my talk, this is a good opportunity to thank our sponsors. Getan, who was sponsored along with FEI and Leica. Without their sponsorship, this event at this beautiful place would not have been possible. I would like to thank everyone for that. And also event supporters, as it's shown here, Gyan, who is basically the initiator for my proposal. Basically, they proposed some global initiative for academic network program, and that's where I started this process. And they gave the funding and ICER, and then it followed with EMBO Global Exchange Series for Professor Baumeister, and then Biophysical Society has given bursaries for around 14 students and postdocs, and Welcome Trust DBT also have very generously contributed to this event, and without them as well this event would not have been possible at all, and I'd like to thank them on this occasion. Good. So let's go on to the talk. So organization of my talk will be mainly hybrid methods. I will talk to you about what is hybrid method. Is it really a single method or a combination of method? Obviously, it has to be combination of method. But can a single method be a hybrid in itself? I will talk about that. What is there in one method that cannot be done by the other? Whether these methods are by itself capable of doing all the aspects of structural biology, or whether they have some particular restriction that other methods can overcome those restrictions. So basically, are these methods competing with each other, or complementary or supplementary with each other? That's what we are going to discuss about them. So whether these methods, for example, now people say Cryo EM will take over crystallography, and crystallography will not have a job, but that's not the situation. The situation is much more profound than that. Basically, they can be very helpful with each other. It can be complementary method, as you will see. I will be discussing about them, giving details of each methods and what are the advantages of each method and what are the pros and cons of combining this method, how we can combine them to get even more information than individual method can give, for example. So let's go on to that. Why hybrid method? Because combining data from many methodologies is very crucial for understanding the functional levels. So for example, crystallography can give static information, so it cannot give dynamic information on millisecond scale. If at all we want to do, it has to be some other method which can give dynamic information. And what are those crucial understanding that one method can give while other methods cannot give? That's what we are going to talk about. And these methods, when they are used in combination, they become very powerful and answer many of the questions that cannot be answered by individual techniques. For example, structure and dynamics of more and more complex systems to be better understood, and their biological functions, and that's the main intention of hybrid method. And for example, you can use a combination of crystallography and Cryo EM to solve even bigger problems that cannot be solved by individual methods. For example, if you want to solve an atomic resolution structure, for example, better than one angstrom, you can in fact do it by crystallography for smaller domains, and then you can incorporate that information, atomic structure, into a global picture of Cryo EM structure. For example, you get very frequently better than 6 angstrom nowadays, and if you can dog these atomic structures, you will get information much more far beyond the information that you get by Cryo EM itself. So that is the power of combination, and each individual technology in structural biology toolbox approaches have its limitations. And what are these limitations? You will see in the image that you are going to see in the next slide. These limitations include the size of the system, the timeline of the motions, the spatial and temporal resolution, and ensemble averaging. So what do we mean by this? It's basically, I'll come back to this slide again. Basically, there are time scales and size scales basically. So various techniques can address problems in various time scales and various size scales, and that is the meaning of that word which we actually. So there are limitations that include for each methodology, and various time scales are also some of the limitations for certain methods, and some of the other methods can resolve those problems. Further, individual methods perturb the system in a way which is very different for very different systems. For example, in NMR, you actually label the atom by isotope basically, and in the case of electron paramagnetic resonance, you basically introduce spin labels to perform EPR experiment. And for example, if you want to do a reconstitution of membrane proteins in micelles, for NMR you basically reconstitute in liposomes. And the introduction of entire domains to stabilize, for example, people talk of stabilizing, solubilizing proteins which are not soluble by adding tags, for example, MBP tags or GST tags. And these are basically, you are perturbing the system to do certain measurements that you can enable that particular perturbed system to use in your technique basically. So in order to crystallize, you add a tag, you perturb the protein, or in order to, for example, in order to do NMR, you basically label with isotope labels, and so on and so forth. So there are other ways of thinking each technique as whether they are physical or radiation method. For example, atomic force microscopy are physical methods. So you actually apply a physical force and then you take measurements, whereas the other techniques like X-ray crystallography and NMR use radiation, which basically interacts with the substance, and then you take their measurements. So in that way as also the systems are perturbed in different way in order to make your measurements of requirement. So each method has its pros and cons, and as systems become more challenging, the perturbations tend to get more drastic and complicated, and the interpretation of these results also become more complicated. So you have to use multiple methods in order to scan the whole range of measurements that you want to do in time and space basically. So this is what is shown here. For example, X-rays and electron microscopy give a static picture, and they range from size 10 power minus 1 to around 10 power 2 nanometer, and EM ranges in a higher range of X-ray crystallography, in the range of 10 nanometer to 10 power 3 nanometer size, and the time scale is very static here in this case for X-ray crystallography. AFM is not included here, which may be in this range basically of the EM, but it's also a static process basically, AFM. So it can come in this range, EM range basically. Minutes and seconds and milliseconds, nanoseconds, picoseconds, NMR can cover dynamic range, whereas the other techniques that cover dynamic range is optical tweezers, single molecule spectroscopy, and FRET is a new technique using resonance energy transfer method. And there are radical footprinting, which uses radiation to release certain side chains of molecule, and then the radical that is formed is being studied. For example, small angle X-ray scattering is another method which uses this range of sizes. So it is in higher range. It doesn't come under atomic resolution range actually. Molecular dynamics simulation, it spans throughout this range. It is computational and also in the time scale, nanoseconds to femtoseconds. As you go higher, it becomes very computational extensive. So this is an example of dynamic macromolecular process that have been measured by various hybrid methods. So in this case, it is a combination of molecular dynamics and Cryo EM. People have got lots of Cryo EM structures of flagellum, and from that they have derived these structures, including nuclear pore complex, which is in the process, and then actin filaments, which is very dynamic process. It is made and broken as and when it's required. And GrowEL is a very classic example for using hybrid methods. For example, people have used molecular dynamics and Cryo EM structures. As you will see, I have given examples in the later slides. This a combination of these can lead to information how the domain movements happen and how the substrate is captured and how the co-chaperone binds, where do they bind, and how the protein gets folded. All this information you can get it through a combination of methods. And this is another example of tomography of HIV with the genome inside. This is the immature virion basically, and ATP synthase. This is a classic example of crystallography and spectroscopy. There is a fluorescence spectroscopy that was used to show that it is rotating as a rotor about its axis. So what they did was they labeled one of the ends of the rotor with an actin filament flagged GFP tag, and then one of the tags, fluorescent tags, they did, and they were able to visualize on a fluorescent microscope that it is rotating when you add the ions. And that was a classic example of a combination of methods. And Johnny Walker got the Nobel Prize for this in 1997, I think. Yeah. Good. So we will go through crystallography and EM. I will compare both of them individually, one by one. So cryo-electron microscopy, as you know, can visualize large macromolecular assemblies, and the size range is going down and down as we are seeing, and resolution range also is becoming better and better as we speak, in months' time rather than years' time. I used to tell people that in last year 2.2 angstrom structure was known, and now I have to tell that it's in last 1 month 1.8 angstrom structure is known by Cryo EM. So information ranges from 30 angstrom to below 2 angstrom resolution for Cryo EM, and the Cryo EM preserves the aqueous environment, and it is done at a condition which is no staining is used. And people talk of this as solution state because it is fractionally frozen at a very short interval of time in microseconds. So you are ideally seeing a solution state. In fact, when you delay this time, you can actually do a time resolved EM map as well. If you delay the freezing from microseconds, 1 microseconds to 2 microseconds at milliseconds, you can actually see how the reaction takes place in Cryo EM as well. So the static process can be seen as dynamic when you do over different time scales actually. And the electron microscopy, as compared to crystallography, is experimentally more amenable, tractable, because you don't need crystals, which is one of the biggest bottleneck in crystallography. And can resolve images or diffraction pattern, and you don't need to worry about phase problem that is there in crystallography. We will see in few minutes when I talk about crystallography. We will see what is a phase problem, and in this case, who discovered this, that phase is available in the images. Can provide 3D volumetric reconstruction information through electron microscopy as it is in the molecule, and suitable for membrane proteins. One of the in the version of 2D crystallography, you can actually solve the structure of membrane proteins. One of the biggest advantages is that membrane proteins do not grow big in size, so they grow usually in lateral size, so it grows in 2 dimensions. So if you have 1 or 2 layers of membrane proteins, which is less than 500 angstrom or 1000 angstrom. It's just the size of the layer of the protein by layer, and you can freeze them, and then you can look at the 2D crystals basically. The aim is to do a diffraction pattern and also take the image of the 2D crystals, and then you have the phases at low resolution and use these phases for computing the diffraction pattern phases basically. Single particle multiple orientation of same particle found in specimen, for example ribosomes, etc., etc. So you get high resolution by means of averaging the various orientations of the single particle reconstruction, and that gives the best resolution among the TEM methodology. On the other hand, electron tomography is a method where you can in fact look at single particles when you don't have models and do an initial 3D reconstruction, or you can look at organelles or the whole cell, for example. The cells cannot be averaged because they are pleiotropic, so they cannot see them. They have to be frozen, and the same cell has to be imaged throughout the 3D tomography method. So you have to use very low dose, and then you have to do the 3D reconstruction to get the cellular section tomogram, which Lugan and Professor Baumeister will explain to you in more detail. If you think of combination of within the EM methodology, if you think negative stain and vitreous ice, if you can do a combination of that, you can have the advantage of both, but disadvantage of the both is also shared. For example, if you do a cryo negative stain, so the resolution is limited by this granule size of the molecules. So we haven't dealt with this, probably I can go through this one by one. For example, in the vitreous ice you have low contrast, whereas in the negative stain you have very good contrast, and the temperature control is not required. You can do at room temperature in the microscope, whereas here the temperature has to be maintained. It's a little bit expensive, the holders are expensive, and liquid nitrogens have to be maintained at liquid nitrogen temperature. There is a risk of high radiation damage because you are actually imaging the protein directly, and in the case of negative stain, it's the die cast that you are imaging, so there is no risk of radiation damage. In fact, I have the grids which I have done in 2007, so we can image it. It's like immortal, making it immortal basically. And image is of actual particle here in this case, whereas it is of the shell. So there is actually, we are indirectly seeing the molecule here, and so there are pros and cons. So when you multiply this, that's what you get when you do a combination of methods. So you can call this as a hybrid within the EM method itself actually. So low resolution up to 15 angstrom, governed by the granule size of the heavy atom that you use, and this you can go up to high resolution better than 3 angstrom. I have put 1.8 angstrom in bracket for Elena. Great choice of initial sample screening, best choice of so this is the best choice of reconstruction because you will get the best resolution, and this is a very great choice. So whenever you want to do, you have to do negative stain first and then go to cryo. So conventional belief earlier it was that the cryo EM cannot go better than 4 angstrom, and you see till 2010 it was a speculation for better than 2.5 angstrom, and resolution, though there were resolutions for virus particles at 4 angstrom, and it was more of a blobology. People used to talk of sausages and helices. Helices was sausages, and then strands they used to term as something else. So it was blobology. EM was a blobology, in fact, basically up to 2010. But as you know, in 2015, Sriram got the structure at 2.2 angstrom, and last month he got another structure at 1.8 angstrom. And incidentally, it was in June again, and this was published in 2016 June, 1.8 angstrom. So every year it's going one angstrom down. So next year probably better. So this is zero point 0.4 angstrom better right now, isn't it? So let us see for the next year at the same time. So this basically he solved the structure at 2.2 angstrom, and he was able to show the side chains very clearly, and you can see the holes in the phenylalanine. That was the so that's what you get to see in a 2.2 angstrom resolution electron density map of a crystal structure basically. So if you go better than 2.2, you will be able to see a dip in the middle of the tryptophan ring and the phenylalanine ring, and we used to get excited when we were doing structures at 1.8 angstrom and see some of these dips actually. And these type of dips are being seen now in electron micrographs actually, electron 3D reconstruction, and that's a remarkable achievement indeed. And moreover, they were able to visualize water molecules as well, and this is a direct indication that you can actually look at the enzymatic activity by using Cryo EM itself. And they were able to do the complex of beta-gal, which is the structure that you are doing in EMAN and other tutorial rely on. So this beta-gal was complexed with a ligand which is shown here, PETG. And this ligand is a breakthrough in visualizing protein-ligand interaction. So this is the example of all the amino acids shown in Cryo EM. So you can see these are the holes that you see at a contour level, 1 sigma or 2 sigma. So you basically see all the amino acids very well actually. On the contrary, when you think of crystallography, I think I should go to a few slides further and then I will come back to this. On the contrary, in the crystallography, you can think of, in contrast with crystal structure of xylenase, which I worked in 0.8 in my PhD. So this is an example of 3 resolution structure at 0.89, 1.1 angstrom, and 1.8 angstrom. So you can see individual atoms separately in 0.89 angstrom resolution. So you can see equivalent to the maps that you saw in EM maps. These are the 0.89 angstrom structure. In fact, you can distinguish here between 2 atoms which are electron dense, which has electron density difference basically. For example, you can see a cysteine has a sulfur which is much bigger than the nitrogen. And in fact, you can differentiate between nitrogen and oxygen also. Oxygen has a bigger density, electron density, so it has a bigger ring or sphere than electron density. So this way in crystallography, you can actually see individual atoms at ultra high resolution. So when you go better than 0.95 angstrom, it's called ultra high resolution. So this is a 0.89 angstrom. You see each and every atom. So this is a comparison of active site tryptophan, which is responsible for substrate specificity in the xylenase. So this is a tryptophan at 1.8 angstrom. You can see it is floppy. You don't see the alternate conformation here at a lower resolution. Whereas at 1.11 angstrom, you see a little bit more orderness, but that is because you can see lower occupancy atoms in this case, okay, because you have data to parameter ratio increases quite a lot at higher resolution, while at 0.89 angstrom, which is frozen, and these two are room temperature and this is frozen. So at frozen temperatures, you can see both the occupancy very well, and one of the occupancy was 0.7 and another occupancy was 0.3. So you get profound dynamic pictures from the static information of crystallography when you go to very high resolution, ultra high resolution. Cryo EM has not reached that stage, so there are advantages of crystallography and Cryo EM, and we have to use them to the best of their capabilities, and that's where crystallography and Cryo EM becomes a very inevitable tool as a complementary method as well as supplementary method. So what is supplementary? I will come back to you soon. I'll go back to this EM example. So why Cryo EM is important? I was talking about chaperones. Oh, this is where I was showing all the amino acids. I think this is not working. I'll put it here and then I'll use this itself. So the chaperones, basically I talked to you briefly in the last talk. This is a classic example for Cryo EM solution state over crystallography. So Chaperones, as you know, are basically a subset of chaperone, HSP60, and they are involved in stabilizing unfolded protein as they emerge from ribosome and folding them in their cage. So there are two groups, 1 and 2. So we worked on group 1, and Helen's group had done lots of work on group 1 chaperonin, and in the Cryo EM structure of this chaperonin, you see a dynamic state in the Cryo EM. Basically, the ADP state has a broader opening in the trans cage, and this is seen only in Cryo EM. On the contrary, in the crystallography method, which was solved at reasonable resolution, you see that there is no difference between the structure of low affinity and higher affinity state. So what I mean is, when they had solved structure of ATP bound GrowEL using crystallography, and they also solved the structure of ADP bound GrowEL using crystallography, and both the structures looked very alike because it is basically crystal packing. In the crystal packing, the cone gets inserted into the base of the other. So the movement is restricted in this case, in both the case of ADP and ATP in the crystal lattice. And because of this, we don't see any conformational change between the high affinity state for the substrate and low affinity state for the substrate. So in the case of natural substrate acceptor state, which is the ADP bound in cis cage, the trans cage is the acceptor for non-native protein. When in the case of ATP in the cis cage, the trans cage has low affinity for substrate, and that's how the substrate protein and other things are ejected from the trans cage when ATP bounds to the cis cage. So this difference in the affinity was not seen in crystal structure, whereas in Cryo EM structure, Helen Ranson and Helen group were able to show that there is a difference in the ADP bound state. When ADP bound is bound in the cis cage, the trans cage is opened and it is more prone for patches that exposes to the hydrophobic residues on the apical domain of the GrowEL. So this basically shows why there is a more affinity for substrate. Substrate bodies have hydrophobic patches on it, and this hydrophobic patches recognizes because it is more towards the substrate acceptance stage. Whereas in the presence of ATP, this is tucked inside and the hydrophobic regions are tucked more inside, and that's why you have low affinity. So this was, they were able to explain through crystallography, and this is a classic example which Helen says that it is more solution state than crystallographic. So that is one more thing. So that was basically the importance of Cryo EM, and now we will look at crystallography and then what is the difference between them, and how these two methods can become a hybrid method or a complementary method very effectively. Crystallography is one of the oldest method, and Cryo EM is blooming now. It's like 1950s of crystallography or 1970s of crystallography is like current Cryo EM stage. Basically, it's developing a lot of developments are going to happen in Cryo EM. But crystallography is one of the very oldest technique, and majority of the deposits in the PDB as of now is crystallography structures, and lots of Nobel Prizes and International Union of Crystallography have dedicated dedicated crystal structure journals now. ACTA D was dedicated for protein crystallography, but they have now started accepting Cryo EM structures as well from last few years, basically 2015 for 14 I think, and methods based on scattering from electrons. So this is a method based on scattering of electrons, hydrogen atoms, usually not seen in this crystallography method. But having said that, if you have ultra high resolution structure, you will be able to see hydrogens as well. Pardon? Neutron crystallography can see that. In crystallography as well, you can see hydrogens, which if you have sufficiently high resolution, that is the data to parameter ratio. If it is more than 6 to 7 times, you should be able to see, and that data to parameter ratio several 10 times probably, I think. If it is better than 0.95 angstrom, you will be able to see hydrogens in many places actually. In fact, modeling, something is missing here. Anyhow, I'll skip that line. Atoms of oxygen, nitrogen, carbon are indistinguishable many a times because the resolution is limited. But as I said, as I showed you an example, the atoms can be distinguished in case of ultra high resolution map actually. And the influence of crystal packing is basically one of the topics that people always deal with, whether it's physiological state or not physiological state. So the standard answer crystallographers have is that you basically crystallize these proteins at a concentration which is physiological basically. So in the cell, the concentration of proteins are almost of the range of 300 mg per ml, and when you crystallize, it is around 10 mg to 20 mg per ml. So it is in the mg per ml concentration is closer to the physiological condition. So in fact, you will have lots of solvent channels also in the protein crystals, and you should remember that whenever you get crystal structure, it is one of the energy minimized state. It may not be functional state, it may be one of the, it may or may not be a functional state, but it is one of the energy minimized state of the protein basically. And it is dependent on so many factors as well. Environment in the crystal is highly aqueous, as I said, water channels, and produces structure in one of the energy minimized state, as I said already. So I'll skip these slides, which I have explained already. So this slide I didn't explain. So this is the hydrogen atoms that you see at 0.89 angstrom. This was a structure of xylanase which I solved in my PhD, and we were fortunate to see hydrogens, and we were able to refine the whole set of atoms using without any restraints. So crystallography, when you have a limited resolution, 2 angstrom, we always use restraints for the bond lengths and bond angles. So in this case, at the end, very few cycles, we released all the restraints and then we refined it. In fact, we were able to identify the difference between glutamic acid and glutamine in that case, basically, because the restraints were released and those two distances were very accurate at this resolution. So how do we use these methods as complementary methods? One of the questions that people ask is, have you shifted to Cryo EM? When I speak to people, because the people who knew me earlier, they know that I was doing crystallography as well. But what my standard answer is, basically we also do Cryo EM along with crystallography. So that's how it is. Basically, you can use both Cryo EM and crystallography as a complementary method, as well as you can use a supplementary. So the input from the Cryo EM, output from the Cryo EM can be used in crystallography as well. Similarly, the output from crystallography can be used in Cryo EM. So that becomes supplementary. It supplies information from one method to another method, and that's a classic example of the hybrid method. So combination of X-ray, protein crystallography and single particle method can tackle complex problems that are impossible otherwise by one method. So if you have native data at very high resolution, say 1.5 angstrom or 0.89 angstrom, and you are not able to solve the structure, and you are able to get a Cryo EM image of this at 6 angstrom resolution or 5 angstrom by Cryo EM, you can use the information the Cryo EM map to face the reflections in the crystallography, and that becomes a very powerful way of getting high resolution map if you are not able to get a Selnameth derivative, or if you are not able to get crystals of Selnameth derivative, or if you are not able to get heavy atom derivatives, etc. So protein crystallography and single particle Cryo EM, basically from atomic resolution small picture to pseudo atomic model. So you are in principle able to get for even more information than the resolution per se can give. For example, if you have a 6 angstrom or 4 angstrom map from the Cryo EM, and you are docking a model which is from crystallography into the Cryo EM map, a monomer which forms a oligomer in the Cryo EM map, then your reliability becomes several fold better than the resolution itself actually of the map. So The range of atomic resolutions for small picture 100 to 200 kilodalton, that is the usual range for crystallography, but there are exceptions like ribosomes and other crystals which are megadalton size. And usually the realm of where the protein does not crystallize, for example higher molecular weight proteins, is of the realm of cryo EM, basically better than 200 angstrom. Now it's reducing as we speak, 135, less than 200 as well. So high resolution subdomain crystal structures can be fitted to single particle cryo maps to get pseudo-atomic model, as I explained just now. So how do we do that? You take a diffraction pattern, you lose the phase information, but you can do those phase derivation either by ab initio method, by crystallography methods, and get the phases. And once you get the phases of subdomains, for example, you get the phases of the one chain of the GROEL, which is a tetradecamer. So GROEL is a tetradecamer. If you see, it is having 7 mer of this monomer on the top and 7 mer of the same molecule, which is around 58 kilodaltons on the bottom. So it forms a double toroid ring basically. So if you are able to solve the individual structure at atomic resolution, and then you are not able to crystallize the big picture of 800 kilodaltons of the oligomer, then you can easily image the 800 kilodaltons and dock this atomic structure into the EM map. So you get pseudo-atomic structure of the protein complex basically. So as I said, we can use the information of phases from the low resolution EM maps to give phases to the high resolution crystallography electron diffraction basically. So can get used to phase low resolution crystallography maps, and once we do that, you have phases up to 6 angstrom for the crystallography diffraction HKL, okay, HKL and structure factors, and you can slowly improve the phase information to the resolution of the data. For example, if it is 1.5 angstrom, you do the phase extension slowly, including step-by-step higher resolution reflections basically. So cryo-electron microscopy and macromolecular crystallography both allow the creation of three-dimensional images of biological macromolecules. And EM data usually provides access to three-dimensional reconstruction of large biological complexes at low resolution. And macromolecular crystallography usually delivers atomic resolution, quasi-atomic resolution structures at 1 to 3 angstrom. So this is a small comparison of if you are using cryo EM and crystallography as individual technique. For example, you need protein crystallography, you need highly ordered crystals, you don't need crystals, you have size restrictions for cryo EM bigger than 150 kilodalton for example, and in the case of crystallography there is no size restriction basically. Generally the smaller the protein size, the better is the resolution, but that's not always the same. Bigger crystals diffract bad and smaller crystals diffract very well as well. There are all sorts of possibilities. The concentration is almost 1000 to 3000 fold less in single particle cryo EM compared to crystallization concentration, and sample has to be homogeneous in the case of crystallization, whereas you can deal with heterogeneous samples in case of cryo EM. And homogeneous sample and cryo EM is the only structural biology method that can provide 3D structures from heterogeneous populations as well. So two limiting factors for protein crystallography which are not encoded by EM is production of diffraction quality crystals and the phase problem. So where do we get phases from the EM? So we have this electron density equation which is basically 1 by V, the volume of the cell, unit cell, summation over all the HKL reflection, and e power f of HKL is the structure factor, which is basically modulus of f of HKL e power 2 pi I phi of HKL. So when you substitute that here, basically you get f of structure factor, basically you get a phase term in this electron density equation. So this phase is not measured when you do crystal diffraction studies. So you have spots, but the phase information is lost. At the same time, when you have an image that is collected on a TEM, you do not lose this phase information, and this was identified first by Aaron Klug. I have a slide for that. We will come to that. It can complete. So electron crystallography is another method where it can complement X-ray crystallography for studies of very small crystals, which is less than 0.1 micron size of proteins, and such as membrane proteins, for example, is the most favorite candidate for electron crystallography. So basically you take out the objective lens and you can do the diffraction, and then if you insert the objective lens, you see the image which has the phase information in it. So that cannot easily form larger three-dimensional crystals can be imaged in electron crystallography, and protein structures are usually determined from two-dimensional crystals in the case of electron crystallography. So you have one layer or maximum two layers, then you will be able to do a diffraction, because if it is thicker than certain range, for example thicker than 1 micrometer, then the crystals are impervious to electrons. So you won't be able to do 2D crystallography if it is thicker crystal. So you cannot do 2D crystallography or crystallography in electron microscope if it is thicker, because electrons interact with the matter and it destroys it cannot penetrate further basically. Electrons can be used in this situation, whereas X-rays cannot, because electrons interact more strongly with atoms and X-rays than the X-rays can do. Thus X-rays will travel through the two-dimensional crystals and without any diffraction significantly, whereas for the same thickness, electrons which interact with the matter can be used as a diffraction radiation basically. So conversely, the strong interaction between the electrons and protons makes thicker specimen which cannot be examined. So there is a problem in that as well. So you have to have a very thin layer. One of the main difficulties in X-ray crystallography is determining the phase. Okay, and this phase is lost in the diffractive image, and because of the complexity of X-ray lenses, it is difficult to image the crystal being diffracted, and hence phase information is lost in the crystallography, as you know. But fortunately, when you do the same imaging in transmission electron microscopy and do electron crystallography, you lose the phases in fact, and then if you insert the objective lens, you get back the phases actually, and that is the phase is present in the image that you collect actually. Fortunately, electron microscope can resolve atomic structures in real space, and the crystallographic structure factor phase information can be experimentally determined from an image Fourier transform. So when you do a Fourier transform, yesterday Elena showed you how to do a Fourier transform of a honeybee, isn't it? So when you do a Fourier transform of that honeybee or whatever the image of the proteins, you still retain the phase information, and this Fourier transform of an atomic resolution image is similar but little bit different to diffraction pattern, with reciprocal lattice spots reflecting the symmetry and spacing of a crystal. So it is a little bit different than that, but it's very similar to that. So typically when you have a EM map, you basically do a molecular replacement with the crystallography data, and then you get the phase information from the oriented electron density map from the EM basically. And this was the main source of this discovery is attributed to Professor Alan Krug, who was the first to realize that phase information could be read out directly from the Fourier transform of an electron microscope image that had been scanned into computer already in 1968. So for this and his studies on virus structures and transfer of RNA, he got the Nobel Prize in 1982. So typically people call this type of phase determination by Cryo EM as by Cryo EM retains the phase. So people like Hobbs, they call it in this article, they call it as EM is a phase measuring diffractometer, or EM images as model for molecular replacement, as I said. So Eleanor has written a paper in ACTAD in 2001 where EM density can be used as a molecular replacement model for solving the structure. So I'll give you some of the few examples before I wrap up. in another 10 minutes. So GROEL by hybrid EM and computational methods gives more profound information as compared to the static picture. So a recent study of dynamics of GROEL, it was published in Cell in 2012, shows flexible fitting of crystal structure of individual components. So you take the crystal structure and do molecular dynamics, and you can do a flexible fitting of the apical domain wherever there is a change in electron density. So that is a very powerful way, and you will be doing exactly the same thing in the last tutorial on molecular dynamics flexible fitting, which Jayati will be taking the tutorial. Jayati and her student basically. A recent study was done, and applying computational analysis to large single particle cryo EM data set. Dan, who was the person who taught me EM and also the image processing and everything related to cryo EM data collection, he was able to determine 6 distinct 3D reconstruction. In fact, I had showed this 6 distinct 3D reconstruction in my last talk as well, but we will go through it quickly. Hence, by going hybrid, one can see large macromolecules in action. So people talk of molecules breathing. So there's a movie. Unfortunately, I could not download the movie, and there was no time to ask Dan for the movie also. So you just see the static picture of that. So he had classified into 6 different classes. Each of them classified, 3 of them in one class were classified as single occupied ones, and the 3 other were classified as double occupied. Single and double occupied is, if ATP is bound to single ring, then it is single occupied. So that is written as S here. And if ATP is bound to both the rings, it is written as D here. So after multiple rounds of competitive alignment and multivariate statistical analysis, they were able to classify into 6 different distinct classes, and truly it's a marriage of flexible bit fitting and cryo EM reconstruction. So if you can actually take the apical domain of the native structure without ATP, and then the crystal structure is known, and then you can do a flexible fitting into this map, and they were able to see a nice. If you go to that paper, you will see. Unfortunately, I was unable to download that PDF file movie. Actually, there's a small movie. If you click on that, you will see that it is moving dynamically up and down. Actually, that whole rotation is being shown. The intermediate domain kinks by 25 degrees, and the apical domain rotates and rises by 100 degrees. Actually, so that whole dynamics can be visualized through a combination of computation and cryo EM actually. So that's a power of hybrid method. You can see the dynamics of the macromolecular machineries as well. So this is another example of a protein EFG into cryo EM density, elongation factor basically. It's a EM density. So the crystal structure, when you look at the EM map, it is very different. So this region basically rotates and gets inside this cavity. So flexible fitting is necessary. So computational molecular dynamics is necessary in order to move this into the density actually. So when these domains were allowed to move flexibly with respect to the rest of the model, a much better fit was obtained. Okay. And this is from the structure of cryo EM structure of EFG, basically elongation factor G. So ATP synthase is another classic example of hybrid method where they could visualize the dynamics of the protein by cryo EM and crystallography combination basically. So analysis by cryo EM revealed the sub-nanometer structure of an intact ATP synthase, and docking crystal structures provided key insight into protein interactions with the catalytic cytoplasmic domain, as well as how these components are structurally coupled to membrane-embedded subcomplex, ring subcomplex. So this also has, there is a very nice movie on YouTube. You can just Google ATPase movie. It runs for 2-3 minutes. You can look at the way in which the proton is pumped from one region to another, and the motor runs, and the ATP is synthesized. So this is the F1 ATPase synthase, and the proton, when it is pumped from outside to inside, it rotates, and the ATP is synthesized here. And this is exemplified in recent work of Liu and Rubinstein of ATP synthase from Thermophilus actually. It was published in 2012 in Nature. This is another example of Cryo EM and NMR, so not only crystallography. So I said at the end I'll deal with a little bit of other techniques as well. So this is a classic example of Cryo EM and NMR combination. So the smaller peptides, which is helical peptides, were modeled by solid state NMR method, and then this was docked into the EM map, Cryo EM map, and to get the overall envelope of the assembly. So these peptides were not crystallized, obviously. So they did this with NMR, and this was in 2014. Small molecule, small angle X-ray scattering is also one of the technique to look at low resolution multi-protein complexes, which are difficult to look at in various other techniques. Probably reveals the shape and size of macromolecules in range of 5 to 25 nanometers. It's rather limited in resolution, but it is very helpful in partially ordered systems, when you handle partially ordered systems basically. So you need the need for crystallization is overcome, and large molecules than NMR but lower resolution can be looked at using SAXS, and this basically leads to hybrid methods. So you can actually dock a crystal structure into the SAXS map envelope, and then you can get a better resolution, pseudo-atomic resolution maps, actually models, pseudo-atomic resolution models. So with that, I think time resolved SAXS, yes, yes, at various ranges. You can see in that plot that I showed in the first. So SAXS scans through certain range, and NMR scans to even more time range actually in that box. Yes. Acknowledgement. As I did in the last talk, Helen, Dan, who taught me the EM, and Helen, who also taught me the EM in Birkbeck, and Art is our collaborator from Yale University, and funding from Wellcome Trust, DBT, and ICER. So when I was in Helen's lab, Wellcome Trust was funding, and ICER funding, and DBT. Thank you.
Regarding the electron diffraction or the electron scattering that you measure, the diffraction pattern, do you also keep an objective and get the projection for the images?
Yes.
Okay,
that's so done. So that's what gives you the phase information.
Okay, because the molecules are stacked upon each other, what kind of projection, what resolution can you obtain from that? Like in single particle, you have a single particle, so the projection would be an approximation of the shape of the particle. In here, the particles are actually stacked together to form the microcrystal. So what resolution can you obtain from the image that you get, projection?
The resolution to which the electrons diffract, so to that resolution you can get, so for example 6 angstrom.
Okay, so would it be as simple as getting a 3D model out of single particle for the small crystals too?
So here getting if you can solubilize the membrane protein in detergents and then get the various orientation, that will be better, obviously.
Okay.
If you are not able to do that and you are able to get 2D crystals, then this will be better. And most of the membrane proteins are very inhibitory to single particle. Basically, they don't solubilize that easily in the liposome as single particles.
So one more technical question. You said the diffraction pattern for electrons can be collected. Can this detected distance be adjusted like in X-ray crystallography?
No. Detected distance is fixed in TEMS.
So that essentially would mean even the resolution that you can collect can be limited. Just curious.
It may be
electron wavelengths are much smaller than X-ray wavelengths. So there's no physical limitation. Only limitation is that the electrons kill very quickly.
Yes.
Yeah, and there's I think amylase or something like that. They have solved those structures at 1 angstrom, 1.2 angstrom using direct methods.
So his question was, in case of crystallography, you move the detector closer to the crystal to get high resolution information. Here you won't be able to. Can we move the detector? Was his question. And
As far as I
that's a
know,
good question actually.
I don't remember how they do it technically, but
Yes.
again, you move it. You move your either your crystals or you cannot move your whole microscope. I guess that would be a bit tricky.
So I think in 2D crystals you don't get diffraction to 2 angstrom and all that. So probably you get low resolution diffraction, very less. So you don't need to move the detector to that extent.
Patterns that show very high resolution data, but I'm not sure how they collected the data though.
Not for proteins.
For protein microscopy.
So
Microcrystals or 2D crystals. There are 2 different directions. One of them is microcrystals, another is one of them microcrystallography, that is 3D crystallography. Another is 2D. There are 2 different things. Are you talking about microcrystals or 2D? They are 1 angstrom, yes. But 2D crystals, obviously, you will have you may have high resolution in 2 directions and very low resolution in 1 direction.
Anisotropic it is. Yes, thickness also. The higher the thickness, then you won't be able to see the electrons cannot pass through it. There is a problem. But thinner, so it has to be thinner so that electrons can pass through it and diffract also. So there is a limitation there.
so I have one.
Yeah, please.
Okay, sorry.
After you. After you.
Yeah, sorry. Since I have also one comment for the, I don't know if I understand the question. Do you ask about the camera lens? If we can change the camera lens or not?
Camera distance. So if you can move the distance of the camera to closer to the specimen, that's what he is asking. Can we move?
Okay, so the so in the electron microscope, my personal, I don't know if trying to answer your question correctly. So we physically the distance is fixed, but since we have a combination of lenses, so we could actually change the distance. Or since by the combination of magnification of different intermediate lenses, we can simulate or kind of change of distance. For example, the first lens, you magnify the previous image about 5 times, and next one change another 5 times. But you can have another combination like the first lens change 10 times, and next one change another 5 times. So its combination become make you much more magnified pattern. And then you have chance to see inner.
I was asking about the electrons that are diffracted from the sample. So after sample, can you use lens to? That's the.
Yeah, it's the
point.
same. Actually, it's the same. They also use. You could zoom in and to zoom out or zoom out to see different details or have different camera lens in the receptor space. Or I don't know, but okay. So I have one.
Rishi may be able to tell us about this. Okay.
Oh, okay. I have a short question, very short question. I'm sorry. And since you have trying to facing the high angle diffractions in X3, so which kind of method did you use for this kind of work when you have some low resolution from low angle reflections for extend your face for high resolution X3 reflections?
What kind of method you?
Yeah. So,
basically, there is a method which Kevin Cowton has developed. So you extend the phases from low resolution to high resolution by iterative cycles. So you add few reflections each round, and then those are included in refinement, and you get the phases of those reflections. When it's reliable, you again add more reflections. So that's how you get the phases from the refinement.
can I consider kind of, algorithm like macro entropy, or I don't know? But
No.
It's different.
Yes. So it's basically
I'm just
you
curious.
have 100 reflections for which you have only 50 reflections phases. So you want to see get the phases for another 50. So you include another 10 in the next resolution bin, the next round of cycle of refinement. So you get the phases for those reflections, and slowly it's called iterative round of density modification and phase extension, basically. Okay. And that's how the algorithm Gary will be able to tell better.
I think that if you have any case everything may work sufficiently. For others, you have to be careful.
Yes.
I mean, I don't know how much time you have to describe all these things. I mean, all density modification work in real space. It means that you add you modify density, and that means that you have a relationship between structure factors in reciprocal space, and the relationship are stronger in shorter range. And if you have, let's say, 3 angstrom, it means that you have a relationship between reflections at 3 angstrom and a 2.99 or something like that. Yes? And you can exploit that as you extend. And you can, in principle, you can exploit that, but I would be careful in general case.
Yeah,
If you have if you can build atomic model, it's a better way of extending phases.
Yes. Good. Yeah. Neil?
I want you to explain the previous question and do it last.
Good.
Do it later. It's okay.
Good. There is one more question at the back. Mike.
It is possible to coat lipid bilayers on EM grids, right?
Yes, yes.
And how do you get proteins embedded into the lipid after you do this? That was a technical question. That's the question I had. If there are membrane proteins with transmembrane domains, after you coat the grid
So
with lipid bilayers,
what they do is they grow the crystal on a dish and then they fish it out, just like fishing out anything. Basically, they dip it and fish it out on the grid, basically on the carbon, continuous carbon. That's how it is done. Chris?
Yeah, I was trying to explain that X-ray and cryo thing, right? So EM thing, electron microscopy. So in X-rays, we cannot deflect X-ray beam, right? So when we get a scattering, it is a cone it goes as a cone. So if we want higher resolution, we want to get maximum angle to be observed. So the only way technically we can do that is to bring detector closer. This is what we do in X-ray scattering. But in EM or electron scattering, you can actually bring that cone of scattering maybe closer by just changing magnification of the lenses, which are electromagnetic
That's what
lenses.
he was trying to explain. Yes.
So I'm just trying to explain like a little better. So
yes.
we really don't need to bring up or down your objective.
That's right. Yeah. Thanks. Krish.
A lot of mechanical drift, which is not very, very serious matter. You can't afford to have drift, mechanically inducing a drift by moving the detectors. It takes some time, natural oscillations to settle down. So that takes a lot of, it has its own problems when you do such things.
Yes.
That's why it's always fixed.
Fixed. More reasons for detector to be fixed.
The diffraction pattern in EM is
yes.
on back focal plane, not at image plane. So that can be also achieved by combination of lenses.
Yes. If there are no more questions.